Dear students, welcome back to the class. As you know that till now I have completed 2 lessons on this chapter, and these 2 lessons it was based on the different terminologies and the very basis of the whole study of inheritance pattern, which we call it as genetics. So I just want to make a small recap of what we have studied in the last 2 classes in a summarized way. In very simple words, this whole basis of inheritance, it is upon the genetic material. And this genetic material, it is enclosed in a chamber which we call it as the nucleus. And within the nucleus, the genetic material which is expressed as gene, it goes from one generation into the other, or from the mother cell to the daughter cells, by a physical carrier which we call it as chromosomes. So the genes, it is linked to chromosomes. So that part, I hope it is clear to all of you. Now there are certain terminologies which are used to study the principles of inheritance, as we are going to study the very basis. Here the genes, it will be called as alleles or factors, and these alleles, they are represented by the alphabets. If it is a capital letter, then we consider that as the dominant allele, and if it is in small letter, we consider that in small letter. As for example, capital T, which is the allele for the character tall, and small t, this is the allele for the character dwarfness, for the stem height of pea plant that Mendel had used. Similarly, this whole pattern of inheritance, mainly it is focused on sexually reproducing organisms. These sexually reproducing organisms, they develops gametes. With the help of gametes, these chromosomes or the hereditary material in individual unit, they come together from 2 individuals, that is the father and the mother, and it is applicable for any kind of living organism, that is a male and a female will be involved here. Therefore, in the resultant zygote, the genes or the chromosomes, they always appears to be in diploid form, and these gametes, they have the chromosomes in half the number, because during the time of formation of gamete, meiosis division took place, and this meiosis cell division, it was essential. Why? Because otherwise the chromosome number, it would have increased day by day, and there won't be any kind of continuity of a particular species or particular race. That is why in first year, in the chapter cell division, the very essence of the meiosis cell division, it was given. That meiosis, it is always required in order to form the gamete, where the resultant cells, that is the gamete, they will possess only half the number of chromosomes. Say for example, in case of human being, if our other cells, they bear 46 chromosomes, that means in the gamete, because of the meiotic cell division, it will possess only 23 chromosomes, and this 23 chromosome from the male and the 23 chromosome from the female, they will come together and they will form the zygote. That means in that 46 pair, in the resultant zygote or in the resultant individual, so that is contributed by both father and the mother or from the male and the female individual. And this condition, it is called as homologous chromosome. That means for a particular character, we have two genes or two alleles. And these two alleles, it can either be in homozygous form or it can be in heterozygous form. Why homozygous? Say for example, if the father have, or if the male individual, it have capital T. Let's take it with the example of Mendel's experiment. If the height of the stem possesses tallness in his gene, and in the female parent also possesses tallness in the gene, that means the resultant plant, it will always be tall. Now, this condition, capital T, therefore it is called as the homozygous one. But in case if the father or the male individual, it have capital T for the stem height, and the mother, it possesses small t for the stem height, and these two alleles or genes, it is going to come to the resultant individual or resultant zygote. But here the two alleles or two genes, they are different. One is capital T, another one is small t, but they are going to express for the same character. So in that case, we will call it to be heterozygous because it possesses two different alleles for the same character. Now, these genes, they are called as the genotype, and the physical appearance, that whatever we see with our naked eye, that becomes the phenotype. We consider it as the phenotype, or the terminology, it is given as the phenotype. The physical appearance, it is called as the phenotype, and the one which we cannot see, that means the gene or the allele which is present in the chromosome, so that is represented with the alphabets, and that is called as the genotype. That means it is the genetic makeup of the individual, and it is applicable for any kind of character the living beings they possesses. So that we have already studied in the first lesson. Now in the second lesson, what it was discussed, it was about the Mendel's experiment. So in Mendel's experiment, what was seen, that he selected 3 steps. Those 3 steps were selection of the parent, then artificial hybridization, and finally self-pollination in the hybrid plants. Selection of the parent, he took 7 contrasting character, and those 7 contrasting character, he made them purely homozygous, and this homozygous condition, it is only called as the pure lines. In case of the pure line parents, Mendel, he allowed self-pollination among the bisexual flowers of the pea plant, which he selected as his experimental plant, and in that way he made all the characters to be homozygous, or it was considered as pure line breeding, which is the first step. Now in the second step, what Mendel did was artificial hybridization. He made one plant male and the another plant female out of these 2 contrasting characters. Say for example, tallness and dwarfness. He made one plant male, another plant female, and in that way he allowed cross-pollination by a method which was called as emasculation. The resultant of the emasculation, it was in the form of seeds, as we know that in case of sexually reproducing plants, the outcome, it was produced in the seeds. So seeds, they were sown, and these seeds, they were all hybrid seeds, and these hybrid seeds, they formed hybrid plant because the cross, it was done between 2 different parent, one with the tall stem height and the another one with the dwarf stem height. Therefore, this is cross pollination. which he had artificially done, and the resultant it was the hybrid plant. And this generation it is called as the first filial generation. Now in case he wanted to see the result for the second generation, in case of that, what he had done, that he allowed self-pollination in the hybrid plant, and in that way he got the second filial generation, and there he got some results. So in today's class, I want you all to learn the crosses properly. Now for performing the crosses, the knowledge from the lesson 1 and lesson 2, it is very much important. So let's start with the monohybrid cross. Monohybrid cross. When a cross is done by considering only one contrasting character, it is called a monohybrid cross. Monohybrid ratio, it is the ratio obtained from a monohybrid cross, and the monohybrid ratio is 3:1. So these are the experimental results of Mendel's experiment. Now let's learn how a cross is done and how do we obtain the monohybrid ratio. For that, two parents will be required which are contrasting in character, that is the tall parent and the dwarf parent, which were made pure line, and now they are ready for the purpose of artificial hybridization. So here capital T, that is the genotype for the character or for the phenotype tallness. Small t, that is the genotype for the character dwarfness. As we know that during the process of formation of gamete, meiosis, division, cell division will take place, and in homozygous individuals, as all the alleles it is same, capital T. So all the gametes it will possess the same type of allele. So in case of the tall parent, below the character tall, as I have written capital T within bracket, and below the character dwarf, here small t is written within bracket. So these two are the gametes. And why I have written it only one T, that is because of the haploid nature in the gametes. And in case of the other capital T, small t, so they were the normal diploid cells. So that is why it is written in 2. Now after emasculation, when they were cross-pollinated or artificially pollinated and they were allowed to fertilize, then the resultant offspring, it bear both the alleles of the parents. As we know that during the formation of zygote, both the parents they contribute to form the character. So in case of this cross, capital T from the male parent and small t from the female parent, they have fused together and they form the zygote. Now this zygote, it is heterozygous in condition. It is heterozygous, but it had shown the phenotype to be tall. Why tall? Because there the capital T it is present. So wherever we find capital T, so that we have to consider it as the dominant character. So as you can see in the F1 generation, or the result of this cross pollination, which will represent as the F1 generation. So this is the first generation. There the genotype it is capital T small t, which is the heterozygous one, and the phenotype that is shown here, that is the tallness. That means because of the presence of a dominant allele, which is capital T in the F1 generation. And all these plants, they will be called as hybrid because the parents, they were totally different. Now in order to see the result of the Mendel's second step, that is self-pollination in the hybrid individual, he had allowed self-pollination in the bisexual flowers of this hybrid plant. And in that, in the second filial generation, he got a similar result. Now let's see what is the result here. On self-pollination of the hybrid plants, it resulted the F2 generation. Now here in the diagram, as you can see, both the parents now they are tall and they are heterozygous. I just want you all to consider one capital T small t as the male part and one capital T small t as the female part. As we know now that on self-pollination in case of the pea plant, as it is a bisexual one, so both male and a female part it will be present there, and since no emasculation is done in this second step. Now the male or the female part, they will bear two types of allele, that is capital T and small t, and this capital T and small t, it will divide equally upon the gametes during the time of formation of the gametes. Now 50% of the gametes, they will bear capital T for the stem height, and 50% of the gametes, they will bear small t for the stem height, and this will happen in both the male and a female reproductive part. In that, now the gametes, when they will come for the process of fertilization, so that is only shown in the Punnett square. If the male gamete possesses capital T for tallness and the female gamete also possesses capital T for tallness, and when these two gametes will fuse to form the zygote, then it will result into capital T small t in the resultant individual. So similarly, if the male gamete it possesses capital T and female gamete small t, then the resultant zygote or the resultant individual, it will bear capital T small t, which will again be heterozygous in its behavior. Then if the male gamete it possesses small t and female gamete it possesses capital T, then the resultant zygote or resultant individual will be capital T small t, which is again heterozygous. And if the male gamete it possesses small t and female gamete also possesses small t, then the resultant zygote or the individual, it will be capital T small t. So this is only shown there in the Punnett square. And on the basis of this Punnett square, Mendel, he did the calculation, and in his calculation also it was found that the ratio it came out to be 3:1. How 3:1? Because as you can see in the Punnett square, we find 3 tall individuals in the F2 generation and only 1 dwarf individual. So in that way, the monohybrid cross, we find it to be 3:1. Phenotypic ratio of the monohybrid cross, it is 3:1. But if we consider the genotypic ratio of the monohybrid cross, then we would find 1:2:1. How? For the genotypic ratio, it became 1:2:1 because capital T, it occurred in only one individual, capital T small t, it occurred in 2 individual, and capital small t, it occurred in only 1 individual. So in the monohybrid cross ratio, the phenotypic ratio, it is 3:1 and the genotypic ratio, it is 1:2:1. Similarly, dihybrid cross, when a cross is done considering 2 contrasting character, it is called dihybrid cross. Dihybrid ratio, the ratio that is obtained from the dihybrid cross is called dihybrid ratio, and it is 9:3:3:1. Now let's see again how this ratio is obtained. Now let's try to understand the dihybrid cross and how this dihybrid ratio it is obtained. So here the parents, here 2 contrasting characters are taken from one parent. Here yellow and round, that means yellow color of the seed and the round shape of the seed, and that is represented by the genotype Y, small capital R, and the other parent, that is green and wrinkled, that means green color of the seed and the wrinkle shape of the seed, and that is represented by the allele small y, smaller. Capital Y, capital R, as this is the dominant one, because here capital letter is written. Therefore, when they were forming the gametes, as we know the gametes, they have the haploid number of chromosome. So here each of the gametes, they bear capital Y and capital R. What is the reason behind it? Because the parental gamete, it is homozygous now. And in case of the other parent, so there the gametes, it will bear small y and small r. And the fusion of these two types of gamete, that will yield the individual which will have capital Y, capital R, smaller. And the phenotype that the individual will show, it will be yellow color of the seed and the round shape of the seed, because the dominant allele it is present there, that is capital Y and capital R. Now when this F1 generation, they were allowed to self-fertilize or self-pollinate, then this Punnett square can be obtained, and this is the probability of occurrence of the individuals that Mendel got in his experiment. In case of the F1 generation, from one individual, 4 types of gametes will be formed, and these 4 types of gametes are capital Y, capital R, capital Y, small y, capital R, small y, and small r. So for each of these character, here also the color of the seed and the shape of the seed, that is only formed, but here the alleles, they may get shuffled during the time of formation of the gamete, and because of that reason, the male part and the female part of the plant, they will bear 4 types of gametes each, and these 4 types of gametes, that is placed in the Punnett square. So therefore we draw this square where 4 boxes are made, and in total it bears 16 boxes. Now if a capital Y, capital R gamete fuses with a capital Y, capital R female gamete, one have to be male gamete, another way have to be female gamete. So that is that the individual capital Y, capital R, capital R. Similarly, capital Y, small r, capital Y, capital R. So that yielded the individual capital Y, capital R, smaller. Then comes the third, small y, capital R, capital Y, capital R. So that the individual capital Y, capital R, smaller. Then comes small y, small r, and that yielded when it fused with capital Y, capital R, this gamete, these 2 gametes when fused, then it yielded the individual capital Y, capital R, smaller. So in this way, just like that of cross multiplication, so we can do it with all the gametes, and at last we get 16 types of individuals, or 16 individuals which is formed by the fusion of these 4 types of gametes. And when we calculate the ratio, then that ratio, it comes out to be 9:3:3:1. Here 9 members, they will have yellow and round seed, 3 members it will have yellow and wrinkled seed, 3 members it will have green and round seed, and only 1 member it will have green and wrinkled seed. So with these two types of crosses, I hope you have got some idea that how crosses are performed. So with this, today's class is ended.